What Keith and myself wanted to do just before we're trying to create a bit of extra space here so that we can go back to the discussion from this morning to finish at least the last two questions around control strategy and risk management and criticality. So we'll try and go through this relatively smartly. So the aim of this was actually to go beyond what we've been talking about for QBD, for essentially immediate release type products, to actually consider the methods of manufacture and how we could become more innovative in that, because a huge amount of what we've been talking about in QBD and the struggles we've had is actually determined by the way and the approaches we take to manufacturing. So there's a lot of new things that are coming through. We're not going to go into a long list, it's not time for that, but just to touch on three of them. We acknowledge clearly that biopharm's in a complicated space, but for immediate release products, relatively straightforward. And if we cannot get our thinking straight around the application of QBD to that, when we come to looking at a more future state around different types of products, then we're really going to have some issues. Now, the challenges to innovation that are often put forward, which are barriers that just need to be knocked over, are things like we don't necessarily have the current levels of technical capability. Well, we need to go out, we need to look at other industries and see if there's things that they're doing that we could usefully use and innovate ourselves. Certainly, there is a need to look at the skill sets that are required, both in industry and in regulators, to look at the new approaches that we're potentially going to be taking. But we have to overcome our inherent conservatism to deploy new technologies when it can really and truly offer us some key advantages. And one of the reasons it's often expressed is that we've got so much investment in the old technologies for manufacturing products in stainless steel that actually we're not going to invest in the new stuff, even though it could give us a step change transformation in the ability to make quality product. There are perceptions of high regulatory hurdles to introduce innovation and a lack of process to manage complex life cycle management changes. We talked this morning about just the issues around non-critical process parameters and the need to be able to change those more simply. But actually, when it comes to more complex changes, there's a real problem in trying to convince people that there's a way through this. And what Keith is going to do is actually look at this and say, well, where are the challenges? Because actually, every time we've seen innovation, we've actually been very supportive from a regulatory perspective. And it's actually to start getting people thinking about, well, what would an overall vision, if you like, to modernize pharmaceutical manufacture actually look like? Now, I don't want to go into all the different types of products that we could be applying this to. There are clearly more complex traditional medicines, prolonged release, more inhaled, things around nanoparticulates that we're using that we need to be looking at from a control perspective. But there are clearly the new novel products, the advanced therapeutics, oligonucleotides, the use of microneedle arrays and things like that. But there I really wanted to concentrate on today with three examples is around more novel methods of manufacturing control in relation to continuous manufacture, something around synthetic biochemistry, and something around discrete manufacture and novel analytics that in GSK, we had a particular issue with a series of compounds in our neurosciences portfolio that were so low dose. We had issues around getting the right uniformity, the right stability, the right bioavailability. We had containment issues, and actually we've come up with a completely different way of making those products, which if you think about the principles behind it, actually might stimulate some discussion. So for continuous manufacture, effectively, we are really trying to look at the things that batch cannot do. Batch is clearly has to be scaled. It gets into all sorts of dynamic issues. We're going to larger and larger volumes. But when you are looking at a continuous process, and particularly with thinking about continuous blending, continuous granulation, it is an equilibrium process. So the scale of scrutiny can be a lot lower, it can be more intensive, and you can apply measurements to how you're actually controlling the flow through that. And as we said, you can run it for a longer period of time, you can increase the flow, but you can actually get more intense levels of control around processes like this. And actually the amount of material that it takes to set them up is actually far less, simply because you run to equilibrium. So you can run for 20 minutes, you can then change the parameters, you can run for another 20 minutes, gain some more product, run for another 20 minutes, change again. And actually you can get a very quick readout on design space and design of experiments to give you a lot of information extremely quickly. But it has got benefits for the environment as well, that we can actually look at making a lot less waste. We can actually work in smaller scales. It is far more scale independent. And there's a business benefit to this. We get more reliable consistency. We get volume flexibility, and it is actually more sustainable, and it's a greener approach. Now, we can apply this both to primary and to secondary, but the controls that are required are definitely different. If we think about secondary, it's very much a case of you take A plus B and you make a physical mixture of A and B that needs some degree of control around it. But in chemistry, it requires a more exquisite degree of control because you take A plus B and essentially want to make C, but don't leave any A and B behind and don't make any D, E, or F. So in generating continuous manufacturing for chemistry, the levels of control around that actually drive us to understanding our chemistry a lot more. In synthetic biochemistry, and a lot of companies are now investigating biotransformations, and this might be an area that we can actually get a lot of learning for the biopharm files and their experience of just how to handle this type of product. But increasingly, the use of enzyme technology is becoming far more into the economic realms of being able to use, even for first generation, enzymes in an aqueous environment to do complex chemistry that actually is difficult to achieve within the labs. It's solvent-free, it uses fewer reagents, and although we're beginning to dabble a bit with individual steps within synthetic routes, there is clearly the prospect of being able to do multiple steps within microbial environments so that actually get a cascade synthesis that could end up making very efficient chemicals for us with stereochemistries that are just not achievable in a laboratory environment. But the one I wanted to sort of major on was this liquid dispensing technology platform that we've got. And as I said, this was for highly potent compounds that had doses that were in the microgram levels in our neurosciences portfolio, and getting uniformity, getting stability, getting bioavailability was difficult. And when we looked at things like spray granulation, we just weren't able to achieve what it was that we needed to achieve. Using micronized material didn't work for us. So we looked into a technology that had been used in other industry and made essentially GMP versions of it to dispense single droplets of drug in solution onto the surface of a tablet, put a lot of controls around it, and actually dry it and pad print it over the top. So you use an inert tablet, and we have actually scaled this to the point where we've got phase 3 and launch capability, as it says on this slide, up to, we have one line that can do 1 million tablets a day, and we could certainly double that on the same equipment to 2 million by just putting filler heads on the opposite side. But what this does, it actually drives a completely different approach that you need to bring to the control strategy. If you think about traditional batch manufacture, whatever we might think about the vagaries of doing sampling and sampling stratification, you do get a result that you infer something about the content and the quality of that batch of material. So sampling is linked to the overall quality aspect of that batch. When you actually make discrete units and every individual tablet is made separately, there is nothing that connects one tablet to the next tablet, to the 10th tablet, to the 1000th tablet. So you need to have a control strategy that actually will look at the quality assessment of every single tablet that you are making. And what we do is we actually take a solution, by and large, or it can be a very fine suspension, but we prefer to have a solution, and we place a single droplet at high speed onto a tablet substrate that then passes through a microwave and infrared tunnel. We do an image analysis of where the drop has landed on that tablet and check it before it's then pad printed. And we have a number of check steps that go in between. The most important ones are we actually use high-speed video to capture the single droplet in flight and calculate through an algorithm the volume of that droplet of liquid. We know the concentration, and therefore we know the dose that's put onto every single tablet. We're actually running this at a speed of about 6 tablets a second, and at the end of a run, we have a complete readout of the performance of every single tablet and what the dose has been that's been applied to that. We also have a check step after the drying stage that actually looks at the position of this tablet. It's not an assay, but it is actually a spectroscopic method to make sure that it's not so close to the edge that it's actually going to be missed when we come to do the pad printing, and we're actually able to make sure that the droplet has landed within the center of that tablet. Now, when it comes to what is the level of performance that we get from this, if we do a comparison against a conventional wet granulated product with 2 milligrams, then at the bottom you've got the traditional plus or minus 15%. And for this product, that is basically a range of batches that we've accumulated together, and it's N of 500 odd tablets, and the process capability is about 1.4. When we actually do the same thing with a droplet dispensing, this is for 600 tablets, then we actually get a process capability of 16. You get an order of magnitude better in terms of the precision and accuracy of the ability to make sure the right dose is on every tablet. Because it's on this surface, we actually have a more concentrated spot, and it's delivered in combination with a film former. So it actually provides us with greater degree of stability. Again, because it's on the surface, we get really good dissolution. So actually, because we've done all the background checks to show how tablets perform with this surface deposition in terms of stability, or particularly in terms of assay and dissolution, then we wouldn't expect to have to follow through with those as actual end product tests. That would actually be real-time release with this type of overarching assessment. So just to finish the overarching control strategy that we have for this, because there are about 5 individual checks on every tablet that comes through this process. We start by looking at the placebo tablet. We make sure it hasn't got any chips or is broken or has got any blemishes on it. We then dispense the solution, having checked the concentration of that solution. We make sure there's a single droplet goes onto every single tablet. That volume is calculated, we know the dose. It's then put through the tunnel where we control the drying conditions. When it comes out of the tunnel, we check the position of that droplet, and then we get 100% inspection after that tablet is pad printed onto the surface. And with that, it actually it's a different approach to a control strategy rather than relying on an end product testing. But because we're not relying on spinning active drug substance around in a big bin with a lot of excipient, it actually lends itself to a lot more at point of use type of control. So I've just tried to use those to sort of stimulate some thoughts around. It is possible to think about different ways of manufacturing products. We are going to see some that are going to become more ubiquitous in what we're trying to do in development and manufacturing. And actually, it will lead to a greater modernization of what we're trying to do in the pharmaceutical industry. That some of our processes, although we've got a lot of stainless steel, is certainly outdated. It's been around for a long time. And it's actually inherently variable, which makes it very difficult to control or to put PAT around it. We need to ensure there's an enabling strategy, regulatory strategy and process that keeps pace with innovation, allows us to introduce this type of approach. But everything that we've tried to tackle, as long as there is the thought around the control strategy and how you get that consistency and reliability, that doesn't seem to be an issue. It is an increased opportunity for scientific dialogue around how you bring in innovative platforms as opposed to just individual projects. I think we do need to think about the skill sets that are going to be needed. Clearly, this goes beyond just having people that know how to make a tablet. It requires people that can do electronics, it can do sort of high-speed, high-quality engineering. The biotransformation is going to look at a whole set of new skills about biochemistry, etc. The enhanced scientific and risk-based approaches to QED actually match this new type of technology, so it actually is giving us a level of control that we're looking for. And as John-Louis said, QED is really around a mindset. It's not just about getting a regulatory file. And actually, if having a simplified manufacturing process can help us to get to a streamlined variations process as well, because there's a higher level of confidence in what we're trying to do, then it's another way of trying to crack this nut about deploying QED principles to our products. So with that, I want to hand over to Keith, who's just going to go through some of the thoughts and the challenges to innovation. Thank you.
Okay, thank you very much, David. David sort of presented some interesting examples of new sort of innovation, new technologies sort of coming on stream in the near future. What I'm going to do, I'm not going to sort of specifically comment on those, but just sort of generally comment on a couple of his slides, really, the challenges to innovation on the desired state slide. Obviously, in terms of the challenges to innovation, a lot of this is sort of common to both industry and regulators in terms of sort of skill set. But just to focus sort of on a couple of these, I think it's important that in terms of the perceptions of high regulatory hurdles. I think there may well be a perception of high regulatory hurdles for some things. But the important thing to sort of remember there is the fact that I think if we sort of are included early in the process, then we can work together to sort of come to a sort of a common position. So so long as we sort of understand what's going on, I'm sure the hurdles are not sort of high. We aim to sort of work with and facilitate industry in terms of innovation. There shouldn't be any barrier to innovation on a regulatory side. Obviously, we have to work within the regulatory system, but certainly it shouldn't be a perception of high regulatory hurdles. We've touched on sort of processes to manage changes. The comment here is for sort of complex life cycle management changes. We've sort of touched on that already. I'll touch a bit on it a bit later. But as I said, a lot of this is sort of industry driven, and obviously within the industry there's got to be sort of appropriate sign-off for sort of progressing new innovation and challenges there. Okay, in terms of the desired state, the vision to modernize pharmaceutical manufacture, obviously the main driver for this is within the pharmaceutical industry itself. But I think as regulators, we've got to recognize that there may well be benefits for a modernized manufacturing process system, which could lead to sort of cheaper manufacture of drugs, which obviously would be a benefit. Enabling regulatory strategy and process that keeps pace with innovation. As regulators, we have to facilitate whatever we sort of are presented with, but acknowledging that we have to work within the existing regulation. So we do have to bear that in mind from the regulatory, but obviously we need to work very closely with industry to sort of facilitate the best we can to enable innovation. The desired state, sort of increased opportunities for scientific dialogue. In Europe, there are sort of various mechanisms for getting scientific advice, be it specific scientific advice or regulatory advice. In terms of the scientific advice, there's a procedure at the MEA where companies can submit their sort of requests for scientific advice. Also, the national competent authorities have their own sort of scientific advice procedures. So you shouldn't forget that, because maybe that would be a useful sort of first port of call, really, to sort of get or alert regulators to sort of something that's sort of in the pipeline. Sort of maybe a bit later on, or exposure to actual working parties, the CHMP working parties, the Quality Working Party, Biologicals Working Party. Certainly in the past, the working parties have been presented with new sort of innovations, technologies, and the example David highlighted there in terms of the tablet sort of dosing one was one which was actually presented to the Quality Working Party. So there are various mechanisms. The PAT team, which I chair, I know a lot of the companies in the audience, represented in the audience, have sort of had interactions with the PAT team in relation to QBD submissions, and we're sort of obviously very happy to meet with industry to discuss various aspects which, okay, I suppose the responsibility of the PAT team is in the QBD area, but if there's something around QBD which is possibly relevant to a new development, then the PAT team is obviously something that an option that could actually be used. The PAT team, just to sort of mention that was actually set up 10 years ago now. There was a comment earlier on about where's PAT gone in all of this? And 10 years ago, PAT was the buzzword. But obviously over the 10 years, the focus and the sort of developments at ICH level, with ICH Q8, Q9, Q10, Q11, the sort of the use of the PAT itself has sort of been lost in terms of the current thing. But as a team, we're still called the PAT team. We could have evolved, I suppose, and called ourselves something different, but we're still the PAT team, but our responsibilities cover anything to do with QPD, design space, the like. Importantly, early interactions is highly recommended. If you've got something in the pipeline, then the sooner you sort of share that with regulators, the best it is, because we certainly don't want to be caught cold by something being presented which we know nothing about. So as I said, early interactions is highly recommended. The education and training and skill sets will obviously need to reflect what the innovation involves, but obviously that needs to also include the regulators as well. The enhanced scientific and risk-based approaches to QBD, obviously the last couple of days has been a very useful exchange of information, discussion around QBD and where we are at the moment. It's obviously been a very honest exchange of information. And in terms of new technology, obviously there's the opportunity for QBD to be applied to whatever is proposed. And importantly, I think we need to apply and build on our existing learning in respect of QBD to do with new technologies. In terms of variations, I'm not going to sort of dwell too much on this because I did touch on this earlier on in an earlier session. Just to maybe highlight, in terms of Europe, we have got a common system now in terms of variation since the August 2013. Prior to that, in terms of variations to national marketing authorization, in a number of member states, they didn't follow this sort of the mutual recognition centralized approach to variations, but now there is a common system which should help moving forward. As I said, I'm not going to dwell on too much on variations. We've already touched on this earlier on. And maybe just to mention again the possible opportunity of using the post-approval change management protocol in terms of facilitating future changes. We've already heard about the global challenges and the differences in the systems across the different regions. I suppose the question is, in terms of streamlining variation process, what do you want? I think the question was asked whether in Europe whether we'd consider possibly adapting the Japanese procedure. I think realistic that's not going to be an option. I think we have quite significant challenges working within our existing sort of regulation legislation, and that would be a huge change, and I think that would, as I said, be unrealistic. So in conclusion, what David and myself have presented is no means a comprehensive view of challenges facing all of us or the possible solution, but obviously we have to recognize that there are always ongoing innovations, new technologies being considered, and the intention of this presentation was to sort of provide a stimulus for further discussion. So the question to the audience is, how do you see the future challenges and opportunities for innovation? Are you aware of anything that should be highlighted to us at this stage? Thank you. It could all be highly secret, I guess, couldn't it?
I just comment, it's Peter Richardson here again from EMA. I think there are any number of things which are innovative, and you could pull out, a long list of things. Maybe some of the things you haven't done too much about there are nanotechnology or other areas you see in the bio or the antibody drug conjugates coming through, etc. So there are plenty of things. But just to mention to you, if it does fall into your area of work, something innovative like that, there is also an innovation task force at the agency, and they also offer you a opportunity to come and discuss how you might see the product being placed, or things that would help you get started on what regulatory aspects may impact on that. Tend to be things maybe more for classification, aspects, maybe for ATMPs, which are a bit borderline, but they see quite a range of different things. So it's just another aspect of what we could offer.
Okay, I think in the interest of time, we do want to try and get back to the earlier discussion. So,
David, it's Frank Montgomery.
yeah.
I was just picking up on some, you mentioned some of the new therapies that are coming forward. You touched on oligonucleotides. We're looking down the barrel of a gun of a lot of mRNA therapies that bridge a number of different sort of standing guidance. But so I think those kind of things, it's gonna build in a lot more cell-based therapies. So I think those kind of task force and groups will be really valuable in terms of how we approach those in the right manner to get those into man in a safe and predictable fashion. So I can see that being hugely valuable.
Yeah, I had a slide, but I took it out, but it was just to reflect on the IMI initiative, the Innovative Medicines Initiative between FPA and the European Commission that has just finished a wave one, essentially, with 2 billion euros worth of investment, and wave two is about to start, and it's really looking at all kinds of new therapies, but there have been requests for projects that have cut across academia, industry, and regulators, and one of the ones that was accepted was delivery of oral peptides and oligonucleotides, and I'm sure you've got people that are connected to that, but those things are going to provide benefit that would come out that will affect us all, and I think that's going to be an additional challenge for us in the future. Okay, thanks, Frank.